of disease, which are disease markers that will help us in terms of evaluating the efficacy of therapies and could be useful as well in terms of diagnostics for NPC. So without much delay, I'll get started. There's an awful lot of research that is going on currently in NPC disease, so this is a very selective view of some of the work, and I've specifically oriented it towards some of the work that I'm most familiar with, which is actually work in my own laboratory. But anyway, I think this should give us a nice point of discussion. And I should also point out that any questions that you have, we'll be happy to take them after each of the talks. But Steve, Denny, and myself will also be participating in the panel discussion from 11 to 12, which we'll hold here. And we'll be happy to take any additional questions you have at that time.
Okay, go ahead.
So in terms of some of the research highlights, I want to talk about 5 areas that have really flourished over the past year in NPC research. The first has to do with understanding the molecular function of the proteins produced by the genes that are defective in NPC disease, and these are the NPC1 and NPC2 proteins. And in particular, there's been a lot of study over the last several years, probably since the genes were identified, trying to understand how they work in cholesterol transport. And I think some of the greatest advance has come really in the past year from Brown and Goldstein's lab in Texas, and trying to understand how these 2 proteins function together in cholesterol transport out of the lysosomes. The next area that we'll talk about will also be the function of the NPC1 protein, specifically in the brain, in the Purkinje cells of the cerebellum. And this is work that still has a long ways to go, but is being pioneered by Matt Scott's lab at Stanford. I'll touch on briefly the therapeutic potential of cyclodextrin in NPC disease, but this is a topic that I'm going to allow Steve Walkley to really expand upon. One of the areas that our lab has been very interested in, along with Bill Walsh's lab from UCSD, has been the analysis of human NPC1 mutations, and also understanding the implications of understanding how these mutations cause disease and how it could lead us in terms of developing new therapies such as chaperone therapy, and I'll talk more about that. And lastly, I'll talk about work that I've done jointly with Denny having to do with identification of NPC disease biomarkers that we feel is very important in terms of allowing us to move forward with clinical trials. So this is a description here, or sort of a schematic of the NPC2 protein, and also of just a portion of the NPC1 protein. These are based upon actual crystal structures that have been generated. Literally, they take the proteins and they produce enough of it, get it in the right concentration and the right solution, and they crystallize it like a cube of salt. And from taking those crystals, they can then do what's called X-ray diffraction studies and can locate every single atom in the protein and can come up with very, very accurate structures. And this has been done for NPC2 protein. It was done a few years ago. Peter Lobel and Ann Stock did this work, and we learned an awful lot from those studies. One of the things that we learned, and I'm not sure how well it shows up here, but I'm going to just take a quick look, is with regard to cholesterol. It's right over here. The cholesterol has a head and it has a tail. And what we learned from their work was that the tail was embedded inside the protein, and that it was really the head that was sticking up into solution. Well, a similar type of approach was recently taken in the past year in the Brown and Goldstein lab, where they looked at the portion of the NPC1 protein and found that it also could bind cholesterol. This was really unexpected. I don't think any of us thought that this region of NPC1 was truly going to be able to bind cholesterol. But what they found out was it bound it in a different way than the NPC2. So instead of having the tail projecting in this case the tail projects out. And it's really the head that projects into the protein, into this binding pocket. So why is that important? Well, it's important because it went along with some biochemical studies that they did in the laboratory that this orientation made perfect sense when they began to consider their model. And the model really exist, that the LDL cholesterol comes in as a big LDL particle. And when it gets into the late endocytic pathway or lysosome, it gets broken down. And one way it gets broken down, do I need it?
Yes.
So I need it, okay. So one way it gets broken down is by an acid lipase that actually cleaves the cholesterol esters to just a free cholesterol molecule. It gets rid of the fatty acid chain. But what's important here is now once it's been cleaved, it can now get bound by NPC2. So we think NPC2 is literally taking the cholesterol that's being transferred from the lipoprotein particle. And then NPC2 is able to basically dock with NPC1. NPC1 is found within this membrane that surrounds the lysosome, and it transports its cargo directly to NPC1, and then it allows for a transfer from the NPC2 to the NPC1. Now, what makes this so elegant is the fact that the orientation of the cholesterol in this binding pocket, as I pointed out here, is perfect for its transfer into NPC1. So it really fits very, very nicely from a biochemical standpoint. And then once this cholesterol is with NPC1, this is where it gets a little bit murky. We don't know exactly how this cholesterol then eventually makes its way out of the lysosome and is able to be trafficked outside the cell. And this is an area that is now going to be, I think, sort of the next sort of frontier in terms of understanding NPC1 biology. But most of the researchers in the field, after seeing the work from Brown and Goldstein's lab and considering the earlier work from Judy Storch and Peter Lobel, I think we feel pretty confident that this makes a lot of sense biologically. And I think this is now where we have to focus to really understand mechanism. The next area that I wanted to touch upon was the work being done in Matt Scott's lab. And this is really work in progress. But what he's trying to do here is to test a hypothesis that rescuing specifically the activity of NPC1 in a very limited population of cells, that is the Purkinje cell neurons in the cerebellum, could improve NPC disease. And the way that he has gone about doing this, he has several people in his lab that have worked on this over the years. Dennis Koh, whose figure I show here, and also he has a student, Mandy Lopez, that's working on this, is to generate chimeric NPC1 mutant mice. And what I mean there is that he's basically engineered a mouse that now has NPC1 expression, but only in the Purkinje cells. And all the other cells in the body don't express NPC1. So he can really look specifically at what that effect is. And what he's found so far is that the chimeric mice have some benefits, but there are still neurological defects, and the inflammation, which is characteristic of NPC disease as well as other CNS disorders, neurodegenerative disorders, that inflammation is still present. And so this raises a number of interesting questions. The Scott Lab has decided to go after it by understanding gene expression patterns. And in these genetic studies, they have seen that when the NPC1 is being re-expressed, it's actually suppressing immune responses within the brain. So this is a very exciting finding, and it would potentially get at some of the questions, sort of chicken and egg questions about what's causing the inflammation, and how, in terms of understanding this mechanism, how we might be able to intervene to be able to prevent some of these inflammatory responses. So this is work that clearly is early on, but I think has the potential to really lead to some important understandings. The next area that received a lot of attention at the ARA-Persegian conference was understanding the therapeutic potential of cyclodextrin in NPC disease. And I show here